Hello everyone, welcome to Global Health Watch. This is Dr. Sean Lin. I used to be a U.S. Army officer and microbiologist and work at the Watery Army Institute of Research. So I'm opening this YouTube channel to focus on public health issues, especially crisis globally. And of course, right now, everybody's attention is on the COVID-19 pandemic. And is the pandemic getting worse? Because so many countries now actually in the hard lockdown, right? United Kingdoms, many of European countries, even Malaysia's and Japan. So are we in a more difficult situation than 2020? Are some of the new variants or mutants more deadly than previous circulating virus strains? Will the vaccine still be effective? These are the key questions people always ask. So in today's program, I'm going to zoom in on one particular mutant. And these mutations happening already emerged in several countries, including South Africa, Japan, Brazil, and China. So this particular mutation is E484K. That's an amino acid change from glutamic acid to lysine at the position of 484 on the spike protein of the SARS-CoV-2 virus. So let's take a look at these mutations. Right, so first I'll show you these slides regarding about the hard lockdown in the United Kingdom, because once the new variants emerge in the United Kingdom, it creates a national crisis, and so already tier 4 level in United Kingdom. And that was actually about that B117 variant. And so this is the UK's B117 variants, and the studies from the London School of Hygiene and Public Health showing these new variants, B117, showing 86% more transmissible than the existing strains. And it already become dominant strain in several parts of the United Kingdom. And this strain already being spread out in many countries as well. And it has been identified in the United States as well. But today we're going to focus on the South Africa strain. And these variants is named 501.v2. And another name is 501Y-v2 that was emphasizing its changed amino acid to tyrosine. So these variants also have a lot of amino acid change, in particular in the spike regions, in the spike protein regions. So it's not exactly the same as the B117, and we will talk more about that. And this 501v2 variant has become dominant in South Africa just in a few months. And so in this diagram, this is a genomic sequencing result, and with bioinformatic analysis for its lineage, for its you can say evolution of this virus strains. So all the green dots representing the variants, and you can see it's a very tight cluster, separate lineage. And then we can also look at the data from South Africa. They also identify. So we can also look at this diagram from Brazil, and they also identify a separate variant. Their variant is named B1128, as shown in green here. And you can see from May to November, and this green, this B1128 variant have become more dominant in Brazil. Right. And we can also see in these diagrams showing the structure of the spike proteins, and you can see these South Africa strain have 3 key mutations in the spike proteins, the E484K, N501Y, and K417N. So these key mutations, key amino acid mutations on the spike protein, it will alter the affinity of these spike proteins binding to the receptor ACE2. And in today's program, I'm going to talk more about this E484K mutant because it also involves immune escape. Right. So when we look at how fast these variants spread, we can also see these diagrams, and this showing the 501YV2 from it emerged in October in Brazil, and it quickly become dominant strain by November 23rd. This is really amazing fast spreading. Right? And also we can see these 501Y2 variants have not only just quite a lot of nucleotide changes in the whole genome, but it also involves many amino acid change. So a lot of the nucleotide change is not synonymous. And also in the spike protein regions, you can see pretty much all the change in the spike proteins is non-synonymous. So it means it can lead to amino acid changes. So this diagram further shows you some of the key mutations in the spike protein of these 501V2 variants. And you can see the RBD stands for Receptor Binding Domain. This is a key domain, right? So in this domain, you have the K417N mutant, you have the E484K mutant, you have the 501Y mutant. So these three mutations we just mentioned, they are all in the receptor binding domain. That's why they can influence how the receptor AC to bind into the spike protein. But at the same time, you see several other important mutants in the upstream, in the N-terminal, as well as some mutations near the C-terminal of the S1 subunit. And so also the famous D614G mutant that's emerged in April and May and become dominant in Europe, and then globally it become dominant. And so basically the South African mutants already have these D614G mutants, and so these 614G mutant is kind of already in the background. With this mutation, the virus has already increased its transmissibility. So on top of this even higher transmitted mutant background, now they create additional mutation on top of it. Right? So, and from this diagram, you can see these mutations, these three mutations become dominant. They almost like evolve together, right? You can see these, the green ones, the light gray ones, the yellow one, the three are the three key mutations we just mentioned. So they pretty much evolve together, and by November it becomes the dominant mutations existing all the sequence they identified. And so these crystal structure diagrams can further show you the position of these key mutations on the surface of or on the interface between the receptors and the spike proteins binding domains. And there are some important mutations in the fusion mechanism, as well as some of the mutation may influence the glycosylation of the spike proteins. So this table shows you a simple comparison of the variants identified in United Kingdoms, South Africa, as well as Japan and Brazil. And Japan and Brazil are relatively similar, and so we put it together here. And so you can see in the spike protein regions, both South Africa and Japan or Brazil variants all have these three key mutations, K417N, E484K, and N501Y. Right, and while the UK variants only have the N501Y. So these make a big difference. And then you can also see these variants also have important mutations or deletions at the N-terminal domain. And these domains mutation may also influence the structure of the S1 subunit, and it can also influence the immune escape capabilities. All right, so we will talk about this later. And this variant has also been recently, last week, just been identified in Guangdong. One South Africa citizens traveling from South Africa into China and also bring the virus into China. That's what the official report states. Right. So now I want to talk about one particular study that's quite interesting. It was done by an Italian scientist, and together with scientists from San Diego and Texas. And so they were studied about escape mutants for SARS-CoV-2 and under the pressure of convalescent plasmas. The convalescent plasmas are those patients' plasma who have recovered from the COVID-19 infection. So they should have relatively good titer of those neutralizing antibody in these convalescent plasmas. So the study design is basically they're using this in vitro assay. So you can have, this is showing you're doing several passages for the virus, right? So you can have different dilution of these convalescent plasmas and put virus, incubated together the virus with the antibody. And of course you can do titering, right? Different concentration of the antibody, or you can say different concentration of the convalescent plasma, and then passage it down for several generations, and then you can try to monitor whether the cells were showing a cytopathic effects or not, right? If the antibodies can neutralize the virus, then the virus won't be able to infect the cells, so it won't lead to cytopathic effects. And so if the cytopathic effects still there, that means the neutralization by the antibody was less effective. So you can titrate it. Right, so this is the general idea of this assay. And the target cell is the viral E6 cell. Right. So in this study, they pretty much follow this neutralization assay for about 100 days. And very interesting, for the first 45 days, pretty much the neutralizing antibody can stop the virus infection. So you can see the titers, neutralizing titers still pretty high. So it means the neutralizing antibodies were very effective against the wild type virus. Then starting from day 45, after that they have a mutant emerge, and this mutant is actually a deletion at the N-terminal domain. This is a deletion F140, and the 36% means if they pick some of the isolate from these different culture plates, then about 36% of the virus, the sequence having these deletions, right? And then by passage 8, so just one more week, these deletions showing the clear advantage, so it becomes 100% showing these deletions. And then if you passage the virus for about 4 generations more, then the very interesting mutation emerged. This is the E484K mutant. And you can see these mutants make the neutralizing titer drop significantly, right? From 640 to just around 40. And then after that, the virus make another dramatic mutation. So this time, the virus put insertion, it's a long insertion of 11 amino acids in the spike proteins. And now, with all these mutations together, it pretty much abrogated all the neutralization activity of this convalescent plasma. The pace of these mutant emerging was pretty fast. It's very shockingly fast. And these diagrams show you these 3 very important mutations locations. So the deletion is at the N-terminal domain, and then E484 is at the receptor binding domain, the RBD domain, and the last mutation emerged is the insertion is also in the N-terminal domain. So the deletion F140 is near the N3 loop. There's a loop in these structures, and the insertion is near the N5 loop. So this table, of course you don't have time to look at the detail, but basically I want to show you just to highlighting that they're testing several convalescent plasma and then identify one particular plasmas. This is the patient plasma 188, and this 88 used to be a very potent convalescent plasma. So you can see the titer is as high as 10,240, and this is very high titer. And then at the last column, you can see the neutralization titer dropped to about 40. So it's hundreds of folds of reduction of the neutralizing capability when the virus have all these key mutations. And you can see compared to the D614G, it's also a significant drop. And the D614G, even though it's shown to have increased transmissibility, but the immune escape capability did not have any dramatic change. So you can see both wild type and D614G pretty much have same neutralizing titer. Right. So this table will show you more in detail about these plasma, convalescent plasma PT188, and you can see how many passages, and you see the change of the neutralization titer at different key time points when different mutations emerged. And by the end, it's non-neutralizing at all, right? So this very dramatic change. And then this study not only used the convalescent plasma, they're also using monoclonal antibody against the virus. And so we can see the last column is the escape mutant, right? So the neutralizing titer also dropped dramatically and pretty much abrogated many of the monoclonal antibodies neutralizing capability. And so if you're using monoclonal antibody, you only target one particular epitope. So it's easy for the virus to be completely mutated. And obviously, the PT188 escape mutant in this particular experiment is very good at evading many of these key monoclonal antibodies that's selected in the study. And those monoclonal antibody primarily targeted against the spike protein, especially the receptor binding domains. And also we need to realize that we need to understand most of the neutralizing antibody are targeted against the receptor binding domains. And also pretty much all the vaccine studies, all the different viral constructs, vaccines or mRNA vaccines, pretty much is targeted against the spike protein, especially focus on developing neutralizing antibody against the receptor binding domain. So now you can see the problem here. The mutations can easily happen in the virus, just about 3 months passages. The virus can develop mutations very efficiently to let them avoid the neutralizing antibody target against receptor binding domain. Right. So this is a big problem. Let's look at more about this study. And this heat map, this is called a heat map, right? So when you have this dark red, it means the titer is pretty high. Right? So you can see the PT188, right? This virus titer is pretty high. That means the neutralization is not effective, right? And this ribbon diagrams also show you the structure change with these mutations, right? So when you had a deletion on the panel A, you see when you had the deletion, the ribbon changes directions, and when you have the insertion on the outside, it also add additional glycosylation site. That's the N248D. This additional glycosylation on this interface. So it will further enable the virus spike protein to evade from the neutralizing antibodies. Right? So these diagram in here, we can take a look at the conformational change triggered by the mutation, like the phenylalanine F140 deletion, and you can see the whole ribbon structure changed, and the direction changed as well. And when you have the insertion on the left on the panel B, you can see the ribbon structure also totally changed. So these deletion and insertion dramatically change the conformation of the spike proteins. Then we can also look at another study. These studies by a group of scientists in Seattle and Fred Hutchinson Cancer Research Center, and they do a comprehensive mapping of the mutation to the SARS-CoV-2 receptor binding domain. See, also about receptor binding domain, because people relatively understand more about receptor binding domain than any other domains in SARS-CoV-2 spike protein. And so they want a mapping of the mutation to see its impact on the recognition by the polyclonal human serum antibodies. So their study find out is the spike protein has 3 regions actually easily to be favor different mutation to escape the recognition by the polyclonal antibodies. So one of the regions is the receptor binding reach, and then there's a loop in the receptor binding motif, and then there's a core receptor binding domains. So these areas showing the red means change in these areas can significantly influence, or you can say change the receptors binding capability as well as antibodies binding capability in these regions. And their study pretty much straightforward, using the E system that can express just a receptor binding domain, and then you screening with different mutation in these East expression systems, and then you testing the antibodies binding capability. So yeah, I won't go into detail about how the experiment was done, but basically through their studies, they identify key escape mutants. So you can see clearly 484 is very outstanding mutations. It happened pretty much very consistently, or for many of the samples that they sequenced, identify. So on the panel B, you can see the percentage, right? And the E484K mutation is 0.11%, and this very high percentage comparing to others only like 0.01%, or so that means this site 484 is so critical for the antibody to recognize the spike protein's receptor binding domain. So a simple change, simple amino acid change the charge in this region. It will change the electrostatics or kinetics for the receptor to binding to the spike protein, or the antibody to binding to the receptor protein. So this mutation is so critical. Right? And this table also shows you, in this particular study, they use 2 antibody cocktail to testing how the virus develop escape mutant. And these one actually give people some hope here, because you can see the first antibody pressure, you can lead to about 31% escape mutants, right? And then when you add second antibodies, right? If you add it separately, the second antibody also by itself about 33% chances that it will develop escape mutant. But when you combine the first and second antibody together, when you make it a cocktail, no escape mutant was identified. So that means the virus had a challenge, or you can say for the virus, it's pretty difficult for them to develop escape mutant if the virus have the antibodies talking to different regions of these spike proteins. So each time probably virus can develop one particular mutants to escape one antibody. So when you have the cocktails, it may not work efficiently for the virus, so it won't be so easy to develop. But I think the difficulty is to give it too much interpretation for this data. I said this study did not give a long-lasting testing or passage of these virus, not like 3 months passage of this virus under the cocktail of 2 monoclonal antibodies. And so it may be just take longer for the virus to generate escape mutant. But in this particular study, they did not do it too long, right? So in here, this is from another study, and this particular study is actually about immunocompromised patient. And this patient had been infected with the SARS-CoV-2, and he has a long-lasting infection in his body, and he was admitted to hospital on and off for several times. And he pretty much was positive for the virus all the times. And they apply antiviral treatment on him as well. And so it's very interesting. They can follow this patient for a long time. It's about 132 days. And so they collecting serum as well as respiratory tract samples from this patient systematically, and then sequence the virus sample isolated from these patient samples. And they tracking the virus mutations from these one particular patient samples. And you can see the virus were able to develop different escape mutants on different time points, right? Especially day 75, and then also day 143, 146. So there are different mutations generated because probably different treatment during this period of a patient being hospitalized. But you can see the virus keep trying to mutate to escape the pressure from antibodies or from the treatments. Right? So this patient was immunocompromised, so pretty much its immune system is weak. The virus have better chance to escape, but even you give them antiviral treatment, the virus still able to escape. And this diagram show you the detail of some of the mutations. And in here, on the lower part, we also see they have these E484K mutation E484. So, and then they also have the 501 N2Y mutants, right? By day 152, pretty much the N501Y mutants all already become dominant in here. Okay, so let's just try to show you there's several studies here, showing you the particular mutants, the E484K mutants, is very easily to be generated by the virus when they under the pressure of convalescent plasma, monoclonal antibody treatments, or antiviral drug treatments. And so that means the virus know what are the key spots to generate mutation to escape all these pressures. And the in vitro study was particularly alarming because it didn't take too long for the virus to find out these mutants that can help them escape the immune system. Right? Only about 70 days they can find it. Just 2 to 3 months, the virus can find these mutants. And we know in South Africa there was a peak in the summertime. And then by November, you know, and these 501V2 variant become dominant throughout the South Africa. The virus, the escape mutant, has such a strong advantage against previous circulating strains, so it becomes dominant very fast. And so we know this mutant has been identified in United Kingdoms, and similar variants being identified in Brazil and Japan now. So it's very alarming. These variants can become the dominant strain globally. And in China right now, we only know that Guangdong identified this one. But how about those cities, you know, in Liaoning, in Dalian cities, and also in Hebei province, Shijiazhuang cities? The whole city was locked down. And tens of millions of people were in a hard lockdown. And the government didn't tell you any particular sequencing result from that region yet. So will there be a very lethal strain, very dominant strain that can escape the immune system's defense by neutralizing antibody? We don't know yet. We definitely want these kind of data to be provided by the Chinese scientists. Probably give the permission by the Chinese government. You know, we hope to see this data as soon as possible. But I think this is very critical information. If these escape mutants exist, it highly suggesting these mutants will be able to evade the antibodies that's triggered by current vaccine designs. All the vaccine designs in China right now, primarily two types of vaccine. One is the adenovirus-based vector vaccine, and these vaccines also primarily focus on the spike protein. And then in China, they also have the inactivated whole virus vaccine. And for whole virus vaccines, usually the neutralizing antibody titer is pretty low. And so when you have lower titer neutralizing antibodies, the virus has higher chance to escape, right? So that's why, even though it's a traditional way to make neutralizing antibodies, but it's not ideal. And then for the mRNA vaccine developed by Moderna's or Pfizer's, the antibodies generated by these vaccines has been shown to be able to target the mutants at N501Y mutants, right? That's a particular mutant that also, yes, exists in the UK, in South Africa, in Japan, in Brazil. But for the mutations 484K, these particular mutants, nobody has given any testing result yet. Probably we will see soon, I believe so, because I think this is one of the most important issue now. All the immunologists, virologists need to quickly address. And if this escape mutant can make the vaccine totally lost its efficacies, then the whole vaccine industry pretty much waste a whole last year's efforts, because you need to probably redesign the vaccines. And of course, you have the production system established, you can change, but all the products that previously produced probably have no use. And then you're also facing another issue with whether the vaccine introduced antibody can generate additional side effects or even more severe antibody-dependent enhancement effects. So I think right now we definitely need to realize these escape mutants, like E484K, and probably with some other deletions on the N-terminal domain of the spike protein, when they work together, these escape variants for the SARS-CoV-2 will make the whole pandemic situations much more worse than it is right now. So I'm pretty much giving a warning here right now. Many people still hope the government can push out the vaccine as fast as possible, right? Hopefully we can get herd immunity, everybody can get protected. But the existence of these variants, these immune escape variants, definitely suggesting this whole strategy may not work. And so I'm definitely very worried about the situation, and I welcome anyone to share your comments, share your thoughts, or share your analysis of different literatures or studies under my videos sections, or the comment section. You can share your thoughts, and I would like to see your input as well. I hope my analysis or my predictions is not accurate, and maybe there are bigger hope. But right now, I'm definitely worried about this particular mutation. And anyway, my first program for my YouTube channel is not so encouraging, but that's a reality. I think that's a reality right now. So I hope you can get some better understanding about the situation by watching my program today. And if you enjoy the program, please subscribe, and I'll talk more about the potential of drug-resistant virus strains in next episode. All right, thank you for watching Global Health Watch. This is Dr. Xiangling. We'll see you next time.